Immunoecology. What does that mean? I think I mentioned this in passing already, several examples, and you all know of this, of course, that whenever a new pathogen strikes a population, almost always not all members of that population succumb to the disease. Some survive because they are resistant or just lucky, and because they survive, the population rebuilds from these survivors. And in the immune system, the antigen presentation process is, of course, a key process, and antigen presentation is efficient from the view of a population if the antigen processing and presenting molecules are structurally polymorphic, so that on aggregate in the population almost all pathogens are somehow presented or shown to the immune system. Of course, the pathogen, remembering our first lesson, the pathogens are trying to avoid this, and there are some viruses, for example, that have means of producing either decoy molecules or downregulating the expression of MHC molecules in the cell, so as to prevent presentation of their viral peptides. A very clever mechanism. Evidence for this interaction between a parasite and a host. But the host, of course, also has to find ways around this, and in other ways perhaps then uses alternative recognition mechanisms, and this is just an ongoing process. But nonetheless, immunoecology is a newly emerging field, and it describes the role of molecules that we are traditionally used to study in the immune system in the formation and maintenance of population structures. And the most famous of these molecules that is being addressed in many studies is the MHC. And you all know MHC is polymorphic, so the blood typing service in St. Petersburg will determine that the MHC alleles that are present in this area are probably quite distinct from the ones that you would find in Novosibirsk or in Frankfurt or in South America, because there is not too much mixture of populations, and populations are being selected based on the local environment, and local environment meaning local collection of parasites. Of course, this becomes less and less important with our civilized states when we move about between different places, but nonetheless there is some adaptation signal that is still visible. So what does that mean? Sometimes there is what one calls the population bottleneck. It can either be induced by infections or parasitic invasions or through some extraordinary event. Think of a major catastrophe, a volcano eruption or earthquake or tsunami or whatever. And what I'm trying to show you is that there are strong pressures in the system that will rebuild MHC diversity to prepare for future adaptation of parasite attack when there is a genetic bottleneck. And this is an experiment here that was published recently. It's looking at the MHC diversity in populations of voles. This is a particular kind of mouse that lives underground and gardeners don't particularly like because they eat their vegetables. So you can see here that what they have studied, number of voles trapped over a period of time. This is years, 2004 up to 2007, and it's even from the back, it's easy to see that the diversity, that the population size, because they're not trapping everything, they're just trapping a selection of these animals, the population is crashing here and coming back again. So something must have happened here. These researchers do not know what happened, but what they observed was that something happened to the population. The population number really went down and then it came up again. Now this was a very good occasion to study MHC allele frequencies. And because there was a significant distortion in allele frequencies over this period of time, they reasoned that there must have been some infectious agent that somehow affected the population. If it was an earthquake, you would expect random loss, because earthquake hits everybody. But if there is a change in MHC frequency, and you can see that here, about equal frequency of two different MHC types, all the details do not matter. Just look at the graphs, about 50-50, 50-50, and then here's great distortion, only 20 or 25% of this white allele, and then it goes back to where it was before. So we're just looking here at two alleles just to make things simple. So this population crash is accompanied by an allelic distortion. And that, of course, is indicative of some selection pressure that was on this particular population. And the selection pressure must have been a positive one for allele A. Otherwise, allele A would not have increased in frequency. Now you might say, if it were very simple and some infectious agent struck, why is allele B surviving? It's a bit strange, no? If we simply associate survival with A, but why is B surviving? This is the answer. B is not really surviving because the only ones that survive in the time of hardship are the ones that at least have one allele of A. This is the double A's, this is the ABs, the heterozygotes, and there is no BB. So in this particular circumstance, having BB is really bad for you. And these animals were completely eliminated from the population. However, the allele did not disappear. So in the circumstance when the population was rebuilding, you can see that it was greatly distorted here. So there was nothing with BB violating the Hardy-Weinberg equilibrium. And then over time, it built back this structure, almost like it was before, because this is what you would expect. No, 1, 2, 1. 1, 2, 1. If there's same frequency, there was about the same frequency, you can predict what the Hardy-Weinberg equilibrium is. This is about right. This is not quite right, but it might become right when you wait a little longer. So amazingly, the population structure in these particular genotypes evened out again. So there was a bottleneck in the population. This one allele was not eliminated, and you could survive. So that means where the selection pressure a different one, you might have observed a loss of allele B completely, and then you would only have allele A. But the prediction would have been that this allele A would then start mutating, and you would build another allele that we would then, in this particular instance, call C. So there is a strong pressure for MHC diversity, even under situations where the population size is very low. Now, when one looks, and what I did not, I think, perhaps precisely mention yet, MHC is not a monogenic locus in the sense that you have only one MHC1 and one MHC2, but we had this example of the Atlantic cod, where they increased or ramped up their MHC number in the class 1 from 10 to 100, I think it was. So a dramatic increase. So we have multiple MHC class 1 and multiple MHC class 2 genes. So you can have, based on their structure, as many as 2 times the number of genes in terms of alleles. So you can go from all genes being homozygous. Say just for the sake of argument, we have 5 genes. You can either have 5 different alleles in these 5 loci, or you can have 10 different ones, and anything in between. Now, what is often observed in populations, and this is an observation that was done by my colleague Milinski, and he was looking at fish here, sticklebacks, in a particular population in northern Germany. What he found was that MHC class 2 alleles for the beta chain were distributed across different individuals, not either all homozygous or all heterozygous, but somewhere in the middle. So there was some sort of optimum that was observed here. Of course, you can also have different haplotypes, just in passing, where normally most of the individuals, for example, in the species have 5 genes, but some might have only 1, or some might have a few more. But we ignore these possibilities at the moment. Suffice it to say that diversity of MHC here often follows an optimum. Now, you might ask, how is MHC diversity maintained? And you have to consider the potential mating strategies. So this is why I was referring to the partying. Okay, so if you go out and try to find a suitable mate, in humans, of course, this is very complicated, but I'll come to this at the end. But imagine yourself being an animal. The only desire you have to manage to pass on your genes into the next generation, and you find a mate that is best suited to achieve that goal. So this is what sometimes people refer to as the selfish gene concept. You really want to get your genes going, the genes take over. The soma is nothing but a means for the genes to be passed on to the next generation. So when you now think about mating strategies, in natural populations, of course not in human populations, but in natural populations, random mating is not a good strategy to maintain MHC diversity. And MHC diversity, because it's very important in regulating pathogen resistance, and you want to be adapted to your local environment, you just go not about and basically take any mate, because you don't have too many chances. You have a limited time for reproduction, you have a limited number of reproductive events, you go through seasonal cycles perhaps, so that you better think twice before you start reproducing, because you want to make sure that all the energy and the effort that goes into this in raising the adult is in the end worth the effort. So random mating is particularly difficult in population structures where there is a low effective population size. What does that mean? Low population effective population size means the number of individuals that I can encounter during my daily walks through St. Petersburg is relatively low. Okay? So there are not very many, in my case females, that would be willing to discuss this particular option. And when you are a lion roaming the Serengeti, you might not be able to find enough females to have a random choice. So when you hit a female, you want to check out that female, not just engage in intense discussion immediately. So if you are thinking about this, you might consider the possibility of directed mating. So not random mating, but directed mating. So this is what you do when you go to the disco. Okay, you have various parameters that you check out, and then after the disco comes another parameter, another parameter, another parameter. So you have a list of things that you go through until you finally hit someone you think, yes, okay, now let's get serious. So directed mating means that you need information. And what is the most important information? Most important information is genetic information. Now, of course, we cannot do genetic sequencing. Particularly the lion in the Serengeti cannot sequence the genome of a potential female lion. That is not feasible to determine whether this lady would produce the most fit offspring if these two genomes were combined. So there must be other ways of working around this. So one strategy you have to adopt is you have to avoid inbreeding at all cost, because that is normally not good. All of us have deleterious mutations in our genomes that, if they were homogenized, would probably cause problems. So you want to avoid inbreeding. So we are not selfing, that's okay. So we are not having that problem. But if we, for example, would select mates from our inner family, this could cause problems because it runs the risk of homogenizing deleterious alleles. So this is what we don't want. So we want to go a bit further. Yet we don't want to go too far, because we want to have genomes that are at least to some degree adapted to our local environment. And if we speak now for parasite resistance, it has to be related to immune genes, and one of the most important immune loci is MHC. But there are other things that need to be selected for. For example, look at me, okay? I'm a multiple mutant. My melanocytes don't migrate properly. I don't really make melanin. It's absolutely appalling. If I were to live in the middle of Africa, I would not survive into a reproductive age. Likewise, one of my colleagues that you might have seen on the first photo, Divine Levere, he comes from Nigeria. If he were to live already as a young man or a young child in the north of Finland, he would have a very hard time making enough vitamin D to survive this environment. So there is local adaptation at all levels, including parasite resistance. So this is what we are being concerned with today. So random mating is not feasible because, as you will realize, natural populations are not very dense. You can get the wrong picture if you go to St. Petersburg or Moscow or New York or Los Angeles. Everybody sits together basically, but that is not normal. Our forebears were dispersed and they rarely met each other. So this is a very low effective population size. So we have to somehow direct our mating choice. Now, information, information, information. So where is that coming from? Many years ago, and maybe some people might have sensed that I'm quite fond of thought experiments, but bearing in mind that what famous geneticists once said, most thought experiments are completely useless. There are some that might give rise to some interesting experiments, and one of the rare occasions I will be discussing now. When we think about our quality control problem, that we are producing T-cell receptors that are, to the first approximation, self-tolerant, so don't cause autoimmune disease, we have to test them for self-reactivity. And I've very briefly alluded to this problem before, without going into any proper detail. But we also recognized from this very short discussion that MHC peptide presentation plays a crucial role in this process. Because MHC presents self-peptide, and this is the thing that the T-cell receptors have to see, and if they do this too strongly, then they somehow are regulated or eliminated. So MHC peptide presentation gives you information about your self, inner self. And your inner self in terms of protein is documented in your DNA sequence. So by reading the protein sequence, you can read, in principle, ignoring now the complexity of the genetic code, but in principle you can read your genetic information. So if there was a polymorphism, and I have another mutation which is in the somatotropin receptor, as you can see, okay, my receptor is very, very sensitive, so this is why I'm growing so fast. Now I've fortunately stopped. So there is variations in protein and variations in physiological outcome. So this is what we are testing, and it's in my genes. If I could read my genes, I could predict what the outcome is, if I can relate genotype to phenotype. And in the T-cell receptor business, we have MHC, this is our mechanism, and we have an organ, the thymus in this case, where this is all happening, but the mechanistic basis is the really interesting one. Now you can make all sorts of rudimentary mathematics and modeling and try to determine what is good for you. How many MHC genes are good for you? For example, if I were an Indian and I wanted to protect myself from the introduction of measles virus by the whites coming into my homeland, I better prepare myself with as many MHC genes as possible to have the greatest possible diversity to be able to present these measles virus proteins to my immune system, and they would kill the infected cells and I would survive. We know from history this has not worked because this population was never under this selection pressure. This is why most of the people died and only few survived. We've seen an example before. So, but nonetheless, the argument could go, as many MHC molecules as possible gives us the broadest possible defense strategy. The trouble is, there's a drawback, of course. And what is the drawback? The more MHC molecules you have, the more self-structures you can present, and the greater the chances that these self-structures hit a T-cell receptor that fits properly and is eliminated from the repertoire. So there is a balance to be struck between MHC gene number, translated into MHC diversity, and the outcome, productive outcome of T-cell selection, somewhere in the middle. And this is perhaps reflected in this picture that I showed you here. it's good to have some sort of optimum. Optimum in the sense of trading reactivity against risk of eliminating too much of your repertoire. And you could do some calculations. This was just to remind me, without going into any detail. You can make all sorts of funny models that would give you a relationship between the number of different MHC molecules and the probability of immune response. And you can read these papers coming from our laboratory. I think it's just a nice mathematical exercise. The numbers are, of course, fictitious in certain ways. Suffice it to say that it makes intuitive sense, and this is represented in this outcome. The number of different MHC molecules is somehow related to the probability of immune response. The more you have, the greater is the probability, but it then starts dropping because it's counteracted by the loss of diversity in the T cell repertoire. Now we do this thought experiment. Now we are again engineers, and we think about MHC peptide presentation in very abstract terms. And I've prepared you for this idea. It's actually a very simple one. And we consider here now just MHC class 1, just to make life easy, but you will get the idea. A protein consists of any number of amino acids that, when broken down by the proteasome, generates peptide intermediates, and we discussed that these peptide intermediates can be loaded onto MHC molecules and thereby saved from further degradation to amino acids. And I've made a very simple cartoon here. So this protein is degraded, for the sake of our argument, into only two peptides, the yellow one and the green one. And because my MHC molecule and the binding groove of my MHC molecule happens to bind only to the yellow one, the green one is degraded and lost, whereas this one is protected from degradation. And only this one then makes it to the cell surface and is seen by the T cell receptor. And this is the logic for the viral escape. So if a viral protein consists only of these yellow peptides, my MHC molecules don't fit. The T cells never see the viral protein. The virus replicates, kills the cell, and happily survives without any activation of T cells. So that is the logic. So now look at this. This is a circle and this is a square. This is sort of an oval circle, but not a square. So this is why I say this structure fits into this one, but this structure not into this one. Now we go one step further, and I hope you will not be annoyed by the simplicity of the argument. Now we do it the other way around. So this is me again. This is Anya. Okay? So same protein, same sequence, same peptide products. My MHC binds the yellow one. Anya's MHC binds the green one. So although we have the same protein, we have different MHCs, and this is why a different subset of these peptides is presented to the cell surface. This is why my T cell receptors are tolerized to this, but not to this, and the other way around. So this is what then gives rise to the mixed lymphocyte reaction. If I use my antigen-presenting cells and use Anya's T cells, they will really go at them because they think they are foreign, and the other way around. Okay, so this is the outcome of this. So the structure of the carrier of these peptides, the carrier that carries them to the cell surface, is mirrored in the structure of the cargo. And now this is the only neat idea in this, that thought experiment. We tend to think of the MHC peptide presentation in terms of MHC diversity, because MHC diversity is what we can determine either serologically or nowadays by DNA sequencing, because we can sequence all the MHC genes and we would immediately see that the sequence of my MHC is different from Anya's. So, and in all likelihood, the binding groove for peptides will also be different. This is the basis of HLA typing that you all heard of. In the old days it was done serologically with antibodies recognizing different structures, but now it can be done by DNA sequencing. So the key insight from this very simple cartoon is that the information about the carrier is mirrored in the peptide. If I now look at the peptide, I can translate back to the structure of the carrier. So if I find these peptides, it's unlikely that this cell or individual has carried this MHC and vice versa. So you're all very clever. You can, of course, imagine this. Now the hypothesis that I made on the back of an envelope was perhaps that is a way of learning about the genotype of a different individual. And there's only one requirement that needs to be met, or rather two. So we have this here. So we have individual 1, individual 2 is the same story. Now it's a slightly different color here, but you can see this oval, you see this square fitting thing. If we organize the system such that it's not like in the MHC cell bound, the peptide MHC complex, but rather a soluble carrier that simply ferries the peptides out of the cell, and then they, of course, they are liberated in the extracellular space. That would then liberate these peptides for structural scrutiny. So if I then had a mass spectrometer, I could take my serum and Anya's serum, and I would find this peptide or this peptide, and I could say, by all likelihood, our MHC molecules are different because they are transporting different subsets of peptides. Or else I could use the existing system, and the only thing I have to do is I have to use exopeptidases and shed and cut off that ectodomain. That's the one that reaches into the extracellular space. This would stabilize this complex and would also liberate these peptides. So the assumption was, if that happens, we can look at the genetic structure of MHC molecules through the structure of their cargo. Okay? Now this experiment that I will be explaining to you in the next few moments started while I was working as a scientist in Cambridge, England, because I had discussions with a colleague who had found MHC fragments in the urine of rats. So he had developed antibodies against MHC, and he was just testing various sources for this, and he found that urine contained MHC breakdown products. Okay, well, very well, that was described, was an observation, but not much notice taken of that. When I thought about this, of course I didn't know about that paper, but then somehow I got hold of it, and I realized that this ectodomain shedding would be exactly what I would have predicted from this very simple-minded thought experiment. So I was really excited about this, and I thought I shouldn't go and find out about the possibility of this process. Now, I'm summarizing this here again. We now know that the peptides bind to the MHC through their side chains, and that the MHC provides a structure, a three-dimensional structure that would accommodate these different side chains. And this is now one particular amino acid side chain, this is another one, and these pockets, of course, are such that only particular side chains would fit. You know all about the details from your textbooks. Now, once again, binding grooves and anchor residues are mirror images. We can genotype this, but we can read the genotype indirectly from the structure of these peptides. And now, how do we recognize things? Through the sense of smell. And the reason why I was interested in the sense of smell, just as an anecdote, was that while working as a clinician, I had to treat and take care of children that had suffered accidents that led to the fracture of the cribriform plate, and that leads to the severing of the olfactory nerves that come from the olfactory system and enter into the olfactory lobes. And that, of course, eliminates their sense of smell, as you can imagine, when the axons are eliminated, but they regrow, finally make contact again, and then they begin to regain the sense of smell. But what was also known was that some people cannot smell particular types of odorants. So they are anosmic for certain types of chemicals, and you might have done experiments in your biology classes along these lines. Also true for the gustatory cells. And what we also knew from the immune system, that some haplotypes in people did not allow them to make antibodies against certain antigens or haptens. So, and that was then later shown to be caused by the loss of particular V regions that we discussed some hours before. So, and then I had this strange idea, okay, well, if that is true, then perhaps these olfactory receptors are also made by the same process that immunoglobulins and T cell receptors are made, by somatic recombination. And if you just don't have to happen to have one particular V region in that thought experiment, you would not be able to sense this particular type of chemical. Turned out that this was completely wrong, of course, because olfactory receptors are quite different types of receptors. There are 7 transmembrane receptors discovered by Linda Buck and Richard Axel, and that project then was finished. I can talk about for hours on this, what a grandiose failure that was. The only thing that was good about this was that I knew about the olfactory system. And Barry Cavern, another Cambridge colleague, whose name should be on here because this picture I have stolen from one of his reviews, was particularly fond of the so-called accessory olfactory system, which is, as the name would suggest, anatomically segregated from the main olfactory epithelium in a separate organ that in humans is called the Jacobs organ, or the vomeronasal system. And this was shown by physiologists to be transmitting information during pheromone recognition. And pheromone recognition, of course, now rings a bell. It tells you this is somehow involved in mating, mate choice, sexual selection, and so forth. Whereas the main olfactory system is more tuned into recognizing volatile odorants. So distinguishing orange from an apple. So, and Barry Cavern, the two of us were sitting in a very small dissecting room, and we were taking out all these things, and I was learning about the anatomy and physiology about this. So he told me that I should try to study whether the olfactory system in one way or another would be involved in recognizing what I perceived as a new type of olfactory ligand, that is these peptides shed from these MHC molecules at the cell surface. He was a very open-minded man. He said, well, that's a possibility. I think it's totally crazy. It will never work. And I had absolutely no idea about the physiology, so I teamed up with other people who could do experiments like this. And that is a piece of result that was coming out of that collaboration with Heinz Brehr and Frank Zufall. What they were able to do, they were able to record activity of sensory neurons in the vomeronasal organ when they were exposed to MHC peptides. And when we saw the first results, we just could not believe our eyes. And this was the result. So we were giving the olfactory epithelium, or we were stimulating it, with peptides that had different anchor residues, indicative of ligands of different MHC molecules. And then we color-coded the response of cells that would respond to one or the other peptide, and you don't have to read the details. Even now you can see green cells and red cells. Green cells stimulated with peptide A, red cells stimulated with peptide B, that were structurally different and characteristic ligands of particular MHC molecules that occur in different haplotypes in mice. And when you overlay them, you see that the picture of activation, or the pattern of activation, is different for this peptide and for this peptide. Some cells respond to both, others specifically to one or the other. And that was a traumatic result, because that would indicate that there are sensory neurons that could read peptide sequences, a bit like your peptide sequencer. Very expensive machines, very insensitive, but these sensory neurons could do the same. And I now show you the sensitivity experiment. Then I go back again. Oh, it's not here. Oh, yeah, it is here. For example, this particular peptide, a very important peptide in the immunological literature, because it's a prototypical ligand that was identified by bulk sequencing of peptides, solubilized from MHC peptide complexes, is recognized at these concentrations, 10 to the minus 14 molar. And you can sit down tonight and calculate how many molecules there are in a microliter, which is the volume of the vomeronasal opening where these rodents take in bodily fluids and evaluate them for genetic composition, as it were. It's basically one cell is activated by one molecule. You can't get more sensitive than this. This is a peptide detector of incredible sensitivity and specificity, as I will show you in a moment. But more importantly, because we are, of course, are biologists, at least at times, these peptides were then shown in a subsequent experiment from the one that I showed you as carriers of that information. Because there is a phenomenon that you perhaps might have heard about, which is called pregnancy block. It's been discovered in mice, again by an English lady, Dr. Bruce. And what she had found is that when you impregnate a mouse, and this is now here a bulb C female impregnated by a black 6 male, this is the blue male here, then, as we've learned, the embryos are heterozygous for the MHC haplotype of bulb C and black 6. And if we now expose this female after impregnation with urine that comes from the father or is supplemented with the peptides of the father, she gives birth to her happy offspring. If we expose this female, having been impregnated by this male with urine that is not of this male's origin, or not of this male's origin, but spiked with peptides of a different MHC origin, pregnancy is aborted. This is called pregnancy block. Quite a traumatic phenotype. So the female senses something changes in the environment. My preferred female has gone. I don't know where it went, but there might be another one now, and this other male is only interested in passing on his genes, doesn't care about the offspring of his predecessor, and either kills the babies when they are born or prevents termination of prevents continuation of the pregnancy and terminates it through neurophysiological means. So the female can read the information in the peptide and in the urine that is in the peptides, and senses now there's somebody that I don't know, some unfamiliar male, and blocks pregnancy. Quite a traumatic result. And that was known for 60, 70 years, but it has been now put down to this molecular information that is carried by these peptides. And the neurophysiological correlate is this one, that these neurons are differentially activated because they can read the sequence. And I have to hurry now. So this is why I just show you one or two more experiments. Of course, you would like to know which type of receptors there are. They cannot be MHC, otherwise this female would not be able to sense the peptides from a different haplotype. So it cannot just generate a haplotype de novo. So there must be another type of receptor, and in collaboration with another neurophysiology lab out of Peter Momberts, we've been able to trace this peptide response to one particular type of receptor. This is now one that goes by the name of V2R1B, and somebody might be interested in these olfactory or vomeronasal receptors. There are complex families of different structures, but there is one receptor here that has this particular name, can be nailed down to one gene, and if you if you, where is it? Yeah. If you eliminate this gene, this one gene, from the genome of these mice, your peptide response that is happily occurring in the wild type is completely eliminated. Indicating this receptor in one way or another is helping to transmit the information of peptides that are activating these particular sensory neurons. Now I have to go back one. You can see what happens when you mutate these signals. This is an MHC ligand that has tyrosine and isoleucine as the anchor residues, where it fits into the pocket of the MHC molecule. When you now take this and mutate it to alanine, and you know alanine doesn't have a long side chain, there is not much that can be considered a side chain, the sensitivity is shifted by 3 orders of magnitude. It just goes right to the right. So the neurons can sense the structure. They can even distinguish. I hope I find that now so quickly. They can even distinguish a single amino acid. Now I have to find this. Now I cannot find that so quickly. So take my word for it. So when you have one peptide, you activate a neuron, and you mutate one amino acid, activation is abolished. So these detectors, these neurons, are so sensitive that they can basically be activated by 1 or 2 or a handful of molecules, and they can recognize a single amino acid change. Again, when you think about it, this is the minimum and rather the maximal capacity for differentiation. And a peptide is distinguished by side chains, and if you change one side chain and you cannot recognize or you have an altered response, that means you are down to the maximum differentiation capacity. Quite extraordinary. So, now what you also see, and you would not be surprised that I show you this, and of course that was my keen interest when we were doing this. Now when we were mutating these various amino acids in different positions, because we know that the MHC ligands have a certain length, and we know that typically the anchor residues occur at certain positions, and we can compare this. For example, class 1 ligands normally have the anchor at 2 and 9, so at the second and the last position of the 9-mer peptide. And then we look at the effect of mutations in particular ligands for the sensory neurons. Here you see them. Which amino acids are recognized when you change them, 2nd, 3rd, and 9th position. And when you look at the frequency of anchor function from MHC peptides, there's a clear correspondence. 2, 3, and 9 are often recognized. Here's the same. MHC class 2 have a different pattern. Most often position 1, 4, 5, and 6 are recognized, and you can see that mutations in these positions also affect recognition. So there is co-evolution. The sensory neuron is tuned into the way the MHC molecule sees these peptides, and then is sensitive to changes in these positions, which makes sense. Or else you could argue the other way around. Perhaps the MHC was tuned into the way these sensory neurons recognized such peptides. We cannot decide. Now, this is the party question. Do humans recognize these MHC peptides? Would that be the explanation for human mate choice? And that is now, of course, the question that interests the tabloid press quite significantly. I don't know what your tabloid newspaper in St. Petersburg is called. I can tell you the name of the ones in Britain or in Germany. The sensational press, the yellow press, that really make a big thing about the most insignificant things in the world. So not a very solid newspaper, but this is what they are keenly interested in. So I was resisting doing an experiment to test whether humans would be able to recognize these MHC peptides for many years. And I had very good reason for this, because I was really worried that this sensory mechanism would be conserved also in humans. And then you can imagine the headlines in the newspapers. So we were working on this secretly, as I would say, for many years, and at that time our children were still at home, and of course they were also beginning to be interested in the opposite sex, and they, of course, that was the sort of dinner time discussions. What are you doing all day and so forth, you can imagine. And once in a while I was discussing that project, and they said, well, why don't you give us these peptides? Can we not kind of make us more interesting to our fellows or fellow female targets, as it were? So they were practicing a bit with this, but that of course is totally non-scientific and the sample size is not good enough. But we wanted to do it properly, and we were testing whether human beings would be able to distinguish peptides of different sequence, because that was basically predicted from the experiments we had done in mice. So would humans be able to distinguish? And would they also be able to produce some sort of more concrete physiological evidence that the brain would somehow react to these peptides? Because psychometric tests are a bit difficult, because you expose people to something and then they have to write down on a piece of paper whether they thought there was a difference here, a difference there, and it's all a bit dubious if you ask me. But we wanted to have at least two components to complete that study, so psychometric tests and this functional magnetic resonance imaging. So what we did was No. Well, you might. So what we did was we provided these probants with a synthetic deodorant. So you can buy them in the supermarket, but we created something that was basically saline and nothing else. And connection lost, huh? Or maybe this is. Yeah, okay. So we gave them basically saline, pretended this was a very fancy perfume, and spiked this with peptides. But before we did this, we tested. Okay, yeah, sorry, I made that mistake. But before we gave these perfumes, so-called perfumes, to the probants, we spiked them with peptides, and we HLA typed each proband, so we knew which MHC molecules it had, and you can look up through the work mostly of our colleague Ramsey which peptides would bind to which MHC molecule. So we would HLA type our probands and then predict which peptides would be their endogenous ligands, and then either spike them with their self peptides or with some other peptides that would not be their own, as it were, endogenous peptides or representatives thereof. And then simply ask them to use that perfume and state on a piece of paper whether they like this odor or not. Because it's very difficult to say, well, I don't like it very much. It's better to say, do I like this better than something else? And this is what our psychologists told us. This is what we have to do. So this is preference for body odor. So we gave them their own peptides, as it were, and foreign peptides, or different peptides. And they, in a complex series of experiments, statistically speaking, they would prefer their own peptides. Sometimes the experiment is a bit difficult because some people carry MHC alleles that we do not know the peptide specificity of, so it certainly helps to bring down the significance of our results. But long and behold, there was a significant difference for preference of self-stimulus when we used non-smokers and people that were behaving properly and were answering in the correct sequence, because it was all very complicated psychological testing. So there was some indication that human beings could distinguish peptides based on their sequence, which I thought was a rather remarkable result. And then we were going on to do this magnetic resonance imaging. And the first amazing result, at least for me, was that when we exposed people to these peptides, more or less a singular region in the brain always responded. Now you have to understand what this magnetic resonance imaging in the functional mode is. You're simply measuring blood flow. And the argument is, if there's a lot of blood somewhere in the brain is thinking. So it needs energy, so this is where the thinking takes place. And amazingly, these people would not consciously recognize what they were smelling, but the brain analysis would indicate that in this right middle frontal lobe, in this particular region here, exposure to peptides would activate this. And it would be possible to repeat this, so self-peptides would always activate that region, which in itself is a remarkable result. So this is a region that is apparently involved in self-assessment. One knows this from other experiments. So this brain region is a sort of multifunctional region. And it would have indicated, if we believe these two types of experiments, that humans are indeed capable of distinguishing MHC peptides. Now you might say, where do these peptides come from? They come and appear in bodily fluids, that is in urine, in sweat, in saliva, in tears. So all that we shed from our body is containing these peptides. So if we make direct physical contact, we are able to take in that information. So this is a remarkable example. Of course, we do not know really whether this is important for humans because we can't do the proper experiments, but there is some indication this is an evolutionarily conserved mechanism. We know this also works in fish, and I can show you, to end the story for today, with a short video about fish that was taken by Manfred Malinsky. You will be surprised how fish can distinguish what they like and what they don't like. So my argument now, and this is basically the bottom line of the story, I think that MHC has not evolved to function in the immune system. Its primordial function was to help in mate selection. Because proteins are always degraded, that is a general phenomenon, and if you then take selective elements from your peptide pool and present it to the cell surface and expose the environment to it, this can be taken as a surrogate of genetic information. So you can learn about genetic similarity or dissimilarity of your potential mate and thereby avoid, for example, inbreeding. You want to establish a certain degree of difference, and then you say, well, that's fair enough, that's probably not my sister, somebody else. And because you are keen on establishing an optimum that is tuned to your original environment, yet diverse enough to be capable of taking on new challenges, this is the way to go for some sort of optimal, complementarity between my own and somebody else's MHC complement. So instead of arguing that sexual selection based on mate choice through MHC was a secondary function of MHC, I would rather submit to you it's the primary function, and only later was it co-opted into the immune system. And why do I say this? I have no idea whether this is actually true, but I think it makes sense when you think about the cell biology of the MHC. I gave you MHC presentation in this cartoon form, and even in the cartoon form it was quite complex. Many components need to be put together to achieve the function. Now imagine you are creating somatic diversification, and you are in dire need of selecting against self-reactivity. There is not enough time to develop such a complex process like MHC peptide presentation. You would be long since dead, and this system would not have taken hold, I think. So my argument is it already was in place, it already worked for mate choice, and then it was co-opted by the immune system to achieve another goal, namely to test androgen receptors for self-reactivity. So this is basically here. There is a question mark, of course, because we are scientists. We will not be able to determine this for certain, at least not in the foreseeable future. But nonetheless, one could at least make the argument that the immune system function for MHC is derived. And now, of course, the question is, because the androgen receptors in lamprey are entirely different from ours, almost certainly their MHC equivalent is also entirely different from ours. So we are now working towards identifying the functional equivalent of MHC in lamprey, and then we can see whether a similar system operates and whether lamprey also respond to, let's call it, genetic information in peptide form. That would be a real turn up. But nonetheless, this is not taken from our own reviews, but that was taken from a review by Wayne Potts and colleagues when they were discussing our results. They think, and I think I would fully agree with them, that these MHC peptides, in terms of providing information not only about protein synthesis in the course of infection or self-reactivity, but also by providing information about the genetic composition, is of course only testing a couple of loci in the genome, and not all of them, but key loci in the genome, to tell the difference between genetically identical or closely related individuals. So in this way, it connects behavior and immunity, and it can guide behavior through mate choice to make sure that your immune system is the best possible one you could generate from the limited encounters you have in finding your mate. So I'll stop here and show you the video, and you will be it's really funny actually. So what Manfred Malinsky has worked on for many years, he has developed the stickleback as a system to test the role of MHC in sexual selection. He did not have a mechanism, but he has simply observed that MHC diversity regulates mate choice behavior. Yeah, so now we have to get that running, and while this is running, I will what's this now? Makes strange noises. Okay. Try again. Well, that's not supposed to happen. Okay, wait. Do something else. So what he's doing, he's basically exposing females, and females, like in humans and mammals, are the choosing sex. So sorry mates, it's not you, it's the females, okay? So the females make the choice. This is why we have to do the song and dance to convince them that we are the right guys, of course. So there we go. Okay. Yeah, this is not good. So what he's doing, basically, he's exposing these female fish to water that comes from tanks where sexually mature males are kept. And these males, they send signals in the water for the female to recognize, evaluate, and then decide, I don't like you, I like you. And they are being exposed into two laminar flow channels, and the female can then just go and swim either on either side, and then the time she spends on this side, sorry, I can't do it now for some reason. So just take my word for it. So the female it's really a funny video. So you see the female from the top. The water flows in this flow channel, and this flow channel, the female can freely move between the two water streams, water columns, and then of course it sniffs it out, tests here, tests here, tests here, tests here, and then it tries to get to the male that gives the palatable water. But it can't do it because there is a mesh that prevents the female from getting to the male. It's just the male water being sent down the flow channel. And you should see the female going crazy when she decided, I'm going for male number A or male number 1. And in this way you can relate genotype of female to genotype of male in a surrogate of these peptides, and you can confuse the male. That's the interesting part of it. So you can simply spike the water and make the water either more diverse and then avoid female or distort female preference. So the female has decided for one male, that looks good, and then you add a few more peptides and all of a sudden the female doesn't like the water anymore because it seems not optimal for her particular own genotype. So I'm sorry I couldn't show you this video, but I might be able to do it tomorrow. I will test it tonight. And I'm finishing here. I've given you these two things in these 2 hours. First, how we work on organ development and providing the morphological basis for quality control at a cellular basis, and at the same time taking that information into some more crazy areas of work that is immunoecology, to understand how mate choice is being regulated and how mate choice interacts with immune system function. I hope at least that I've convinced you in the last part that this is an interesting area of work. There is many questions that we haven't addressed yet and which we are completely ignorant about, but at least there is some indication that there is information transfer from a system that seems seemingly confined to the interior of an organism, but this information is passed on to the outside world, can be recognized and evaluated by other individuals, and that has significant implications for the population structure. So enjoy the evening, and I'll hope to see you tomorrow again.
So you said, if I'm correct, that the human experimented subjects, they preferred the self-order in a way, right?
They prefer?
They prefer the self.
No. Only in this particular experiment, the psychology was to ask them, which perfume would you like for yourself? Okay. And when you think about it, it's again for the boys. If you are fond of a particular girl and you go and buy them perfume, a waste of money, because your preference is not hers. So the only thing you have to do is you have to go and sneak into her bathroom and check out what she is using and then buy it. But don't trust your own taste, because it will not work. By definition, it must be different. Okay? So just as a piece of advice.